Welcome back. So we are going to focus first on lab biotechnology and talking about gel electrophoresis and polymerase chain reactions. So we'll start with DNA sequencing, and this is when we image DNA strands to compare similarities and differences between different samples. So we can take the DNA of one animal or one human or one plant and compare it with another one to see which different parts of the code are similar or different. And this is commonly used in forensics. So if you ever want to become a detective, they utilize a lot of DNA sequencing to figure out who done it and who had the crime or who committed the crime at the crime scene. So they would take the DNA of what is seen at the crime scene and the DNA of the individual that they suspect to be the culprit and compare them. And if they're the same, they look the same on the gel electrophoresis, they tend to be the criminal, obviously. So we're going to watch this quick video that explains how this works.
DNA fingerprinting uses a number of molecular biotechnology techniques to determine the genetic profile of a DNA sample. If the sample is too small, PCR may be used to increase the sample to a workable quantity. Treat the DNA with restriction endonucleases to splice it into fragments at specific locations, known as restriction sites. When one enzyme is used, it is called single locus probing. With more than one enzyme, it's called multiple locus probing. The DNA fragments are separated by size using gel electrophoresis. A DNA marker of a known size is often added for reference. Perform southern blotting by applying a nitrocellulose filter to the gel. The DNA fragments are transferred onto the filter. Radioactively labeled DNA probes are applied to the filter. Any probe complementary to the DNA fragments will hybridize, and excess probes are then washed away. Sandwiching X-ray film on top of the membrane will reveal any hybridized areas on the film because of the radioactive labels in the DNA probes. The X-ray film is the result of the southern blot technique and is called an autoradiograph. The pattern on the autoradiograph is the DNA fingerprint and can be compared to other DNA fingerprints from separate samples to detect a genetic match.
So that video went a little bit more in depth as to what I explained. I don't expect you to memorize any of these specific techniques or what a restriction site is and the single locus technique. We don't need to know that here for this class, but I just thought it was a nice little video that kind of takes it one step further if you're interested in learning more about the lab techniques. So they alluded to PCR in the video, but we can also utilize PCR to help amplify our DNA. So let's say that we don't have very much genetic material available. We can undergo a process called polymerase chain reaction that will amplify the material, the genetic material, to then be ran through gel electrophoresis. So in the next video, we're going to learn a little bit more about how PCR polymerase chain reactions work, and then we'll move on into reproductive biotechnology.